Hi, welcome to the Bioinformatics chat. Today I am talking to Sergey Nurk, who is a postdoc at the NIH. Hi, Sergey.
Hello, nice to be here.
And Sergey Koren, who is a staff scientist at the NIH. Hi, Sergey.
Hey, thanks for having us.
And we will be talking today about genome assembly and about the classic assembler Canu. But before we get to the topic of the genome assembly, this is the first time I have anyone from the NIH on the podcast. And so I'm very curious, guys, if you could talk about your experience working at the NIH. How did you end up there? What are you doing there? And how it's different from working at a normal research institute or a university?
So this is Sergey Koren. I can go first, I guess. I've been at the NIH for about 5 years now. And I ended up there together with Adam Philippi, who is our group PI. We were actually at the same place together. I worked with him for almost 10 years now. And we transitioned together to the NIH to start the research group that's there now. Before that, we were at the National Bioforensic Analysis and Countermeasures Center, which is hard to say, but it was based in Frederick in Maryland, and it was primarily a lab for the FBI, run by as a contract for DHS, the Department of Homeland Security. And it kind of provided the support for investigations. So based on the original Amerithrax mailings, when someone mailed anthrax powder to the senators and other people like in the media, the government decided that they needed to have capability to analyze this in-house because at the time they had relied on Tiger to do the analysis. And so we worked at this lab, we were doing technology development, so seeing how best we could analyze these types of data, but also providing support for actual ongoing investigations. And we were there at an exciting time because they had the first PacBio sequencer, and we were working on making that useful for microbial data because that's obviously of great interest to this kind of forensic lab. And as the instrument kind of got more and more mature and we started sequencing things like goats and cattle with our collaborators at the USDA, we figured the Department of Homeland Security is going to frown on us using our resources and time to improve agricultural products and not necessarily related to biodefense. And so when this opportunity in the NIH came up and Adam applied for it, it seemed like a great opportunity to continue our research with long reads with maybe a little bit more flexibility of topics. And the NIH has been really great so far. I mostly have experience with NHGRI, which is the institute we're in. I can't speak for the other institutes, but I know that NHGRI has been extremely good. They're very flexible about the kind of work we do, even though we're the human genome research, and we certainly do human research. In terms of genome assembly, we also have a lot of freedom to work on other projects. We've had the same USDA collaborations. We've had this large collaboration with the Vertebrate Genome Project working on assembling all the vertebrate genome species. And NHGRI has been supportive of all those projects. And, in terms of, I haven't had as much experience working in a university as a PI or as a professor, only during grad school, but, certainly there's less pressure than NIH to be submitting grants and writing grants. We do have reviews by external investigators who come in, and you have to give a research proposal and the plan, and they evaluate what you've done over the past period and what you're planning to do, and give you a rating and say whether you're doing science that is worth continuing to fund or not. But it's, a lot less intense, and there's you spend less time preparing for that than you would in terms of writing grants, I feel like, in a university as a professor.
It's Sergey Nurk now. I can say from the postdoc's perspective, so far it has been great. I also don't need to apply to any, additional grants, and I can focus on my work. And Adam, as a PI, is great, and he gives a lot of freedom and a lot of, opportunity to choose what projects you are interested in, and you can collaborate with other people in the group and other people in the institute as much as you think is productive for your research. And our institute and our lab in particular are part of big consortiums. So we are in the midst of it all. So I think Serge can tell you more about his work on VGP consortium and others later, if you're interested, and of application actually of Canu and Canu to those consortiums as well. But I also wanted to say that probably compared to like a standard university, there is maybe more bureaucracy, I think, in NIH, because it's governmental institutions, and I had to pass through all different security checks and so on, and I was waiting for my laptop for 2 months. But then, coming from Russia, I'm pretty used to bureaucracy. So as long as you conform to the rules, you're good. And the IT has been very supportive, and we have amazing computational resources that we can use on a daily basis without almost any restrictions, which has been very, very useful for my work so far. So, and there is also a huge community of postdocs from all around the world. NIH is a huge, so NIH in Bethesda has a huge campus, and I know that I don't know, there are like thousands of postdocs, and they are organizing different events and going to museums or to the concerts or just to the restaurants or organizing some lectures. So it has been fun.
Awesome. So let's talk about Canu. And Sergey, Koren, you were one of the original, I guess, authors of Canu, but also Canu has an interesting story coming from the Celera assembler. So can you retell that story?
Yeah, so the Celera assembler kind of came out of Celera Genomics, which was the company that Craig Venter founded to beat the public effort to sequence the Human Genome Project. And so it was really one of the first assembler designed for whole genome shotgun data that could scale to these large eukaryotic genomes. So the first paper describing it was actually this Drosophila paper explaining how they assembled the Drosophila genome and scaffolded it with Sanger data at the time. When Celera essentially largely shut down, and most of the people who had worked on the assembler left and went to the J. Craig Venter Institute, which was a nonprofit research institute, they ended up open sourcing Celera assembler, and so it's been kind of open source maintained since then. I came into, when I originally switched to bioinformatics from doing software engineering and kind of traditional working at Amazon, etc. type work, I joined Tiger, which is the Institute for Genome Research in Rockville. And there was a group there which worked on genome assembly because Tiger had been doing a lot of assembly and finishing of microbial genomes and had moved into the space of doing mosquito and other genomes. When I joined, the first project I worked on was actually a tick genome, which was one of the worst genomes ever to work on. And partially we found out because they had actually sequenced there were two different tick colonies from different parts of the United States that they crossed in a lab and then sequenced those, a whole pool of them to get enough material of DNA, which no one considered, who had planned the sequencing. They didn't think about the fact that we're introducing a lot of variability in the tick populations, and that's all reflected in the read. So it was really hard to do a reasonable assembly of that data. But this was kind of my first exposure to bioinformatics. And so there were two separate groups at the time when I started. There was one group in Tiger, which ran the Celera assembler and then made contributions to the open source. And then there was a group at the J. Craig Venter Institute, which had a lot of the alums from Celera actually, who worked on, more algorithm development and other features. And shortly after I joined Tiger, it merged with JCVI and it became just JCVI, J. Craig Venter Institute. And so the two groups merged, and I started working more directly on the Celera assembler. And one of the first projects I worked on there was adapting it to the new era of short reads, which were 454 reads at that time. And so it was kind of a fortunate time to join bioinformatics, because it was the time when I remember when I first joined Tiger, we had these meetings where it was kind of like, well, should we get 7x coverage or 8x coverage? We would save $100,000 if we did 7x. Right. And all these projects were kind of like, yep, we're going to get 7x. Maybe this project will push it to 6x because we want to save money. But then this whole field got opened up in a huge way by the short read era and all the sequencers coming out from Illumina, 454, Solid, all these different options that hadn't been available, that made the tools have to change, and that I was fortunate enough to work on several of them. And so I worked on the Celera assembler and at JCVI on adaptations to it for the 454 data, for using it to finish genomes easier, which was a thing people did at the time, which people would now, no one would do, but we did still at that time. If you had a microbial genome and you had some gaps, you would do primer walking into the gap. And so then you want to, when you're assembling, that those reads should go into the middle of this gap, right? And so you can localize that in the assembly algorithm and constrain them. So those are some of the early things I worked on with Celera assembler. And then I got lured away to this National Biodefense Center that I mentioned before by Adam, with the promise that there was this sequencer called the PacBio, and they had the first commercial installation of it, and they wanted to use it, and no one really at the time knew if it was going to be useful or not, or how useful. And so it just seemed like it had a lot of promise. I had seen presentations of it because they had come. Stephen Turner had come from PacBio to JCVI to try to convince JCVI and us to adapt Celera assembler to that data. At the time, it was really early data. I think it was maybe at best 1,000 bases long reads with 30% error or so. And they had this thing called strobe reads, which were kind of like paired end reads, but more than two pairs. So I got lured away by the promise of working on this. And that was when I first adapted Celera assembler to these long reads by doing this kind of hybrid assembly where you corrected the noisy data from PacBio with more accurate data from Illumina or 454. And then we contributed that back into the Celera assembler, which was at the time still primarily maintained by JCVI. And later on, a couple of years later, the opportunity came to hire one of the main engineers from that group, Brian Wallens, into our lab, which we did. And that's when we kind of set him to the task of throwing away all the changes in Celera assembler that we didn't really need, the things that have been adapted to 454 reads or to Illumina reads, or even old Sanger support, like scaffolding Sanger data. And we set him to throw all that away and clean up into the third version of Canu, the CA started 3G was the original name. And so we decided we needed something more pronounceable than the CA 3G. And that's when we came up with the name Canu. And it's essentially been surprisingly successful. I've been surprised by how popular it has been. But that's
so how did you come up with that name?
So we spend an inordinate amount of time coming up with names. Actually, it's kind of a shameful part of how long we think about our names for our tools, given how bad some of them have been. But we had this PBCR, which is a terrible name, I'll admit, or PacBio to CA, which was the name for our original correction process. And we didn't want an acronym because they tend to be very hard to pronounce and not very easy for people to remember. And so we were thinking of, at the time I was also working with our colleague Todd Trangan, who's now at the University of Rice, who had been working on other projects and was interested in the shipbuilding and kind of how you do like overlapping structure of the ship when you overlap the wood boards. There's different ways to construct ships where you either overlap or abut the wood and things like this. And so, overlapping assembly. And we decided to go with a kind of boat theme, and we wanted to name it a canoe. But we were afraid that canoe is not a very easy to spell word for non-English speakers. So we decided to just shorten it and make it, the phonetic version of canoe.
That's a smart move.
Yeah. And it has the advantage that it's uniquely spelled, so it's easier to find than if you had actually named it the proper canoe, right?
Yeah, that's a good move. The only name for bioinformatics software that I've ever come up with was SPAdes, and it's not super easy to Google.
Because it's also a game, right?
Because it's also a card, and it's also a small shovel.
Right, yeah.
Wait, are you saying you were the one who invented the name SPAdes?
Yeah.
Wow, that's another story that we definitely will
Yeah,
hear later.
we also spend a lot of time picking out the name, and yeah, I can tell you later. But yeah, I think I was the one who suggested SPAdes.
Wow. Okay, so and then the Canu was born. Was it true that from the start you were targeting specifically the long reads, or at least as long as they were at the time? So on the one hand, you were using the Celera assembler, or the at least the general framework from it, and you were targeting the long reads. So was there any particular reason why the ideas behind the Celera assembler were well suited for long read assembly, or was it simply, a historical sort of accident that was what you were familiar and that was what you were working with?
I mean, it was partially that we were familiar with it, and the ideas are kind of applicable because if you look, a lot of the assemblers that have been developed for the long reads, with the exception of Fly, have followed the same string graph or overlap graph approach, which is what Celera assembler was originally built with. And so the Celera assembler actually slightly predates the string graph, which is why it uses overlap graphs instead of string graphs in its implementation. But that idea is kind of nicely suited to the long reads because you don't have too many of them, and so you can actually look at graphs of where your nodes are the reads, and without too much redundancy, right? Like you would get within a Illumina dataset. And you have this nice feature of tolerating errors because in the Brown graph, which is what people had traditionally been using for the Illumina data, every error you have kind of adds noise to your graph, right? Because it doesn't have an overlap, and so you have to process the reads carefully to correct them, and all these other steps. And we could just kind of say, well, we don't care about these kind of small differences. These reads are still coming from the same locus of the genome. So it kind of naturally fell to being applicable to the long reads because they were essentially Sanger reads plus, right? And so we were always targeting my focus, at least, has always been on the long reads. Celera assembler the J. Craig Venter Institute group did spend time trying to adapt it to Illumina data, but they were never very successful with it, just because of the volume of data. It just doesn't suit itself well to this overlap graph concept. So I think it was much better suited for the resurgence of these long reads that started with PacBio, but then certainly also when we were developing Canu, we were very aware of the nanopore reads, which were just becoming more popular and more applicable to larger genomes. And so we were definitely targeting those two as our primary use cases.
I think that makes a lot of sense, but could you introduce our listeners to the concepts of overlap graphs and string graphs?
Yeah, and I mean, Sergey, feel free to jump in too, because you've been thinking about these graphs a lot recently as well. The basic idea is that in the graph is that each read is a node. It actually has two nodes, one for each end of the read. And so then you can have other read ends attaching to the 5' or the 3' end of a read. And so this ends up as a bidirected graph where you can essentially choose to walk a read in any direction with a constraint that if you enter through the 5' end, you have to exit through the 3'. You can't enter and exit through the same end of the read. And
so just to clarify, the reason for that complexity is that you don't know from which strand the particular read comes from, right?
Right. Each read could have been read from an arbitrary strand. So you have to, and it's a mixture, right? So you may be walking a path where one read in your path is from the forward strand, the next read is from the reverse strand. So you have to be able to switch strands as you're going along in the graph. And the big, I guess, distinction from the Brown graph, which you're probably familiar with, or your listeners are, is that the reads are kind of atomic units. We don't split them up like you would in the Brown graph into the k-mers. You just look at the entire reads. And in the overlap graph, you always keep the read full length. So when you find the branch and you end up at a repeat, you end up with this kind of non-intuitive assembly where you extend into the repeat by the length of the read that's on the end because you don't break that read. And that's the kind of the model that Celera assembler has been using, which is sometimes good, but sometimes confusing for downstream users because they see that two contig ends have an overlap, so they want to merge them, but they may not be mergeable. It's just that both reach into the same repeat, but that doesn't mean that they are the only ways to reach into that repeat, for example. And so that's where the Brown graph and this A Brown graph, which is what Fly is using, have an advantage because they bluntify the ends, and so does a string graph tries to bluntify the ends, so that you end right at the start of the repeat. You don't extend into the repeat by a read length.
Actually, I'm not sure that originally. So yes, a string graph is a very messy notion, I think, because it has been different people consider different things of string graphs. So Gene Myers, who introduced them, didn't make it easier for people because in his own paper, the description is slightly different. So of how to get string graph slightly not maybe contradicts, but does not very well correspond to the picture that he is giving to illustrate them. So while illustrating them, he shows something more similar to these Abraham graph, repeat graphs that Misha Kolmogorov and Pavel Pevsner are using now. But in the description, the string graph you get from overlap graph by just removing transitive overlaps. And then in the description in the paper, Gene Myers actually says that it's so obvious that this gives you the same essentially graph as the De Bruyn graph would give for the same set of reads if they're perfect, that it doesn't require any further. So it's so obvious that you don't even need to describe it further. It is actually not true, I think. But where are we? So Serge told you what repeat graph is, and so in our at least in our group, we call by string graphs, they're not bluntified usually, but they are the overlap graphs with transitive overlaps removed.
So can you explain it further? What are transitive overlaps?
Sure. Yeah. So if you have three reads, A, B, and C, and A overlaps with B, then B overlaps with C, and A overlaps with C, and the size of the overlap between A and C is well corresponds to what you could infer from the other two. So from A B overlap, and BC overlap, you can try to infer what overlap between A and C should be, right? Given the length, that the length of the reads and the overlaps. And if the overlap that you have in the graph actually corresponds to what you would infer not having it explicitly, you just remove this overlap. So it simplifies the graph quite a lot, and then you actually indeed are getting these long uniting paths, that everybody wants to get, in which you are certain that they are part of the genome, right? So then we're coming to this notion of what are unitigs, what are contigs, and so on. But if you then the very basic trick is to start extracting the non-branching paths in this graph.
Right, and that's part of the reason of transitive reduction, right? That you're removing redundant information that would otherwise look like a branch.
Yeah.
Because otherwise it would look like you have a branch from A where you could go to B or C. Yes, I guess in my mind, I think of the overlap graph as the non-bluntified version, the string graph as the bluntified version, only because most of the assemblers that have called themselves string graph, starting with Nubler for 454 data, which was written by Jim Knight, try to bluntify the ends and break at the repeats, as well as Falcon, which was developed by Jason Chin at PacBio, also bluntified. So I usually say overlap graph for non-bluntified and string graph for bluntified, but I agree with Sergey that the paper notion, the string graph paper is not super clear about what exactly the string graph is itself.
Right, and go into more modern software like the software by Heng, Lee, right? Miniasm, I think he also says that he uses string graph, right? As it is described in the paper, but it doesn't do any bluntification, I guess.
Maybe also let's explain in more detail what bluntification is.
Yeah, sure. It's what Serge mentioned before when you end up with overlapping unitics. So you do not try. So essentially, one region of the genome can be represented by several different overlapping contigs, which is not always what you want. And actually, compared to the graphs that Fly gets, and compared to De Bruijn graphs, your graph ends up being dependent on the set of the reads that you were provided with, rather than only on the structure of your genome. So essentially, if you have enough coverage, De Bruijn graph, and you don't have errors, De Bruijn graph will always be the same for the particular genome. It's not like that for these non-bluntified string graphs, which can be different. Yeah, but I mean,
so when you bluntify, you basically remove those overlaps?
Well, you can't remove them from all of the. So you need to pick a node which will still have this sequence, right, and remove it from others. But it's hard to do in so. Since there are, cycles in the graph and things like that, it's hard to come up with a deterministic rule that will do it well. And we actually gave it some thought, and it can be formulated as integer programming problem, I think, but nobody has done that. So people usually do some heuristics to do that. And also, I don't know, but I think. If I remember our discussions correctly, Nubler also tries to bluntify so that it would detect the repeat boundaries, right? So another problem is if you just bluntify, the result still wouldn't give you the precise boundaries of the repeats that you actually might want to know. And this is another, upside of using De Bruijn graphs, repeat graphs, that you immediately get the boundaries of the repeats, which actually can be convenient for using some additional information later on to, figure out what's going on. So as a representation of the genome, I think repeat graphs are in many senses better, but the assembly topic is a very different one, and string graphs and overlap graphs are still in many quite convenient for the assembly, because they give you very good control over what overlaps you are considering and which ones you are filtering out. So if you would we talked that you can just extract the unitics from the overlap graph that you got, right, from the string graph. But the problem with this approach is that they will be much shorter than you would like them to be. And they will be much shorter than what any modern assembly software will give you. So then starts the ugly part of heuristics of how you make them longer and what overlaps you can ignore, what reads you can ignore, how you can resolve the genomic repeats, which are essentially longer than the minimal overlap size parameter that you set up. So,
right. And so maybe it's a good time to mention that actually, even though we've been talking about string graph and overlap graph, Canu, by inheritance from Celera assembler, actually uses a slightly different concept, which is the best overlap graph, which is a very simplified form of the graph. And so this was first, again, proposed and introduced for 454 data. But the idea is that you only allow a single edge from every end of a read. and that's its best edge. So best you can define in different ways, but traditionally it's defined by the number of matches
within the overlap, right?
Yeah, or the length of the overlap, something like this. But it has several advantages. One is that your graph is very small in memory. Another is that you don't have to do any of this transitive reduction or other simplification steps, because you can't have an edge from A to C and to B, right? Because A will say, well, I have a bigger overlap to B, so it implicitly reduces the transitive overlaps just by that filter. And this is a pretty, simple, greedy heuristic, but it does still build branching paths. So it's not like you'll always have a non-branching path, because you'll still have a read that says, well, A is my best pair, but A may say, well, C is my best pair. And so you end up with this branch where B wants to go into A, but A wants to go to C. And so within Canu, what we do is we build this best overlap graph. And there's a lot of filtering up front, because as Sergey mentioned, it's really important to, you have fine control of what overlaps you keep. And so you want to get rid of as many false ones as you can, where false ones here would mean ones that originate from other repeat copies, right? So anything that comes from a different locus of the genome. So if you can tell that this overlap between two reads is from a repeat and induced by a repeat, and not actually from two reads that came from the same locus of the genome, then you want to just ignore it. That'll make your assembly much easier, right?
But we don't ignore those. No, we ignore the reads which are incorrect. But I think the problem, so we would have fewer problems actually if we really considered this graph. The problem is that we don't even look at the branchings at this graph, but we try to. So yeah, what Canu does, I mean, I wouldn't suggest anybody to use best overlap graphs ever, because they have interesting problems, and actually your unities are not guaranteed to be parts of the genome if you just look at the best overlaps. But right. Canu is even messier, and so it extracts long greedy contigs and then tries to bring overlaps back and try to see whether it just traversed some of the regions of the genome where it actually didn't have the information to do that. So whether it, resolved some repeats incorrectly. So it tries to bring this information later and break the initially constructed contigs. So this filtering that Serge mentioned is we mostly need it for the initial contigs to be as correct as we can, although they are super constructed in a very heuristical way. So it's a mess, and it's a legacy that we live with. But for everybody who is coming into assembly, don't do that.
I think given that
there are
the number of reads and the length of the reads, you can build the string graphs or overlap graphs.
Yeah, so now the cool thing about assembly now is that there are some algorithms and modules that you can reuse. So if you have the reads, you don't need to construct if you have the overlaps, you don't need to construct the string graph yourself. You just take mini-asm and put the overlaps there. And there you go. You get some approximation of what you want, and then you can try to start on top of that. But in the times of Celera, there were no such software, and also there were no big enough machines to put this entire graph into a single node, and so they really had to be very minimalistic with the memory requirements. So that's why it uses this design, but we have so much headache with it. You can't imagine.
Yeah, I think, that's a good point, that building an assembler is a relatively complex undertaking, right? There's a lot of modules and things. That's getting easier because, like Sergey said, there's some reusable pieces now you can put together. But because of that, there's things in Celera Assembler or Canu that like this use of this module that builds this best overlap graph called Bogart, which, we haven't been particularly happy with, that we were working on replacing, but then it takes time because you you move one step at a time. So first you improve the previous step, then this step, then this. And so you kind of slowly are continually refactoring bits as you get better ideas or get better approaches and things like that. And it often takes longer than you'd like to get all those things rolled out into the assembler version. So you kind of work with what you have and improve it as you go.
So while we are on the subject of like the high-level concepts behind assemblers and like different paradigms, Sergey, Nurk, I wasn't originally planning to talk about this, but I just cannot pass the chance. So yeah, why don't you tell us also about SPAdes, which is an incredibly popular assembler. So what's the story behind that, and maybe how does it compare to Canu in terms of its paradigm?
Well, I think the only way it compares to Canu is the messiness of the code base at this point. Although my former colleagues are doing their best to, bring it to the modern software standards, and I would say that they are succeeding. So SPAdes started in 2011 when Pavel Pevzner, my former scientific advisor, he got a big grant from a Russian government of the Russian Federation to establish a modern bioinformatics lab. I was one of the original hires of that lab, mostly because I knew the guy who was organizing it. Then I left my job.
And that was in St. Petersburg?
Yeah, in St. Petersburg. Yeah, I left my job as a software engineer in Yandex and joined the world of bioinformatics. And
Yandex, by the way, for those who don't know, this is like the Russian version of Google.
Yeah.
Russian version of Google++, because they also have like the Russian version of Uber, right?
Right.
I think they have more services than Google does even.
And they also bought the Uber in Eastern Europe, I believe, recently. So anyway, and that's what Pavel told us, to develop a genome assembler. So there were some other projects as well, but there were many of us, and I think he thought that it's a nice way to, occupy many of us. And also he was hoping that we would come up with some, maybe new approaches to genome assembly. Spoiler alert, we did not. And but the motivation behind all this was that at this point, Pavel was very excited about the new technology for sequencing genomes of individual cells. And the motivation for this, so it's single cell sequencing, but right nowadays everybody thinks about single cell RNA-seq, right? But for us, it was single cell DNA sequencing. And the problem there was that for many microbes that are around us, they're hard to culture. They're hard to cultivate. So we know that there are like in any soil sample, there are like thousands of different microorganisms. But when you start trying to get the colonies, only like a dozen grows or something. So a very small part of the entire diversity. And so single cell DNA sequencing with and then Roger Laskin, I think he was actually employed by JCVI.
Yep.
At that point, he came up with this method called MDA, multiple displacement amplification, to try to amplify the amount of DNA that you get from essentially a single microbial cell, to then allow the standard sequencing to be applied. And it worked. But the problem was that the resulting data sets, they had some weird properties. First of all, the coverage was super uneven, and many places of the genome were not covered at all, could have been missing. And this really messed with the assembly software that was available at the time. And another problem which aggravated the first one was that from the method itself, it resulted in formation of the chimeric junctions. So some of the sequences, some of the reads that you saw were actually joining distant parts of the genome, but the difference, for example, from chimeras that you usually see in PacBio and that Canu tries to avoid by pre-filtering of the reads, is that in MDA, the same chimeric junction ended up being supported by multiple different reads. So because it was actually, formed as a molecule and then sequenced. And so you had to come up with a heuristic to deal with those. And just filtering by coverage didn't work because your coverage was very uneven. And so some of these chimeric junctions ended up with higher, read coverage than other correct genome fragments, just because, those were so low covered and this chimeric junction formed early in the process and ended up with high coverage. So, and we came up with some methods to deal with those, and we were actually very much inspired by the work of our colleagues from Hong Kong, who were developing IDBA assembler series, and in particular IDBA UD. I think it's an amazing piece of software which is, I think, being used by this day by many labs. But we also came up with some other methods based on, the network flows, arguments, and estimation of the multiplicity of different regions of the genome, even without being able to rely on read coverage to do that. So and then we continued working on it, and we were working, and it started becoming popular and was faring well in the independent benchmarks. Serge, were you the co-author of this Mazurka benchmark that really, spiraled us to fame?
Yeah. So I worked on the Gauge paper along with Adam, who did a lot of the actual validation for that paper as well. Yeah. That was primarily led by Steven Salzberg.
So I think the idea was to promote the Mazurka assembler, but the byproduct was that SPAdes was faring very well in most, comparisons. And then, since it was benchmark used by the authors of Mazurka, everybody was like, oh, okay, probably we should try using SPAdes now.
So I think the original idea was to promote an alternative to the Assemblathon series, because that had been the initial benchmarking. And so given the experience of, everyone in Steven's lab and nearby in terms of Mazurka and having people who worked in Celera assembler around, Steven had an interest in having an alternative to Assemblathon that was more focused on, I guess, reproducibility, where it's not, because in the Assemblathon, the first Assemblathon, people just submitted assemblies. And so it was a bit hard to know how they were produced. And so the Gauge benchmark, one of the main ideas was that they're all run by the same person. So it's, what do you get if you run someone's assembler? Certainly if, I'm running Canu, I can do better than pretty much anyone else running Canu because I know all the heuristics and tricks and tweaks. Right. But if someone takes it, what do they actually get if someone runs it without having that familiarity?
And I think it was a great idea. And also I think Assemblathon was focused more on vertebrate genomes, right? And Gauge was like the first, I think, initiative to try to provide provide the comparison of microbial assembly methods. And I think nowadays there are similar projects live on. I definitely remember there was a project based on BioBox's container which was, so there was at some point a website where you could see the latest versions of assemblers, tried on different microbial data sets. I think it's very useful, and it was super useful for us as developers to see different metrics that people, really care about, because we were so we had some notion of what we try to do, but then it's another. It's very refreshing to see what other people care about and what actual, downstream analysis needs, because we were not working so much with the biologists as much as we wanted, I think, and as much as we needed. So, yeah, it was really cool. And then different spin-off projects started, and there was this, metagenomic version that I was very deeply involved with, and there was like RNA-seq version of SPAdes, and all these different like SPAdes for plasmids and so on and so forth. Nowadays it's I think that I know the analogy, actually. The good analogy I think of SPAdes and Canu is that it's a huge code base. And so for people who know them, right, for the developers, it's a very cool playground. Because it's the same with Canu. So with High Canu. So whatever I wanted, I was asking Serge and Brian how we should implement that. And, 90% of the times they told me that it's already been, something very similar has already been done, and we can just try to reuse it and adapt this module. And I was like, oh, okay, cool. And that's what, made the progress very fast. I think it was the same with SPAdes at some point. So we had so many diverse, modules, implemented, that, adaptation for each particular, problem or new data type, we could do it fast. It wouldn't mean that we could do it best. So, specifically designed software or algorithm targeting particular problem and particular data type would probably do better than us. But then we could prototype fast. And, it's as always, like the simplest tricks give you 90% of the results. And then you spend the rest 5 years pushing towards the final 10%, which doesn't always work in the sequencing and in bioinformatics world, because in 5 years, the technology is obsolete and nobody cares anymore. So in some sense, it's not very cool as, it's not very, scientific maybe, but it ends up being very important to be able to prototype fast if you want to keep up with what's going on, and to keep producing useful software.
Yeah, I mean, I think that comes back to some of the points that, Sergey had brought up before about how parts of Canu are messy, like the best overlap graph. And if we were making a new assembler from scratch, we might not use that. But because, we have optimized it somewhat with heuristics and other things, and it works quite well for the current data type, it works, and you can prototype, and then our focus gets diverted to other parts, like working on making it applicable to hi-fi data or other kind of things, just because there's a lot of code in an assembler that is hard to go through and replace. And there's not many assembler projects that kind of survive for a long time, right? I mean, SPAdes is one exception, but as Sergey said, there was a pretty large team working on this. And there's often an assembler that's developed, and I've been guilty of this myself too during my PhD, where I developed like a scaffolder that then I never maintain, and so no one's going to use it because I haven't made any updates to it in years.
And I've tried.
Yeah, exactly. And so it's easy, it's easy to prototype something quick, but to make it survive for many years, you naturally build up this big code base, and some things you're not happy with that you're always trying to improve. So I think that it's kind of a double-edged sword. The reason that Celera Assembler and Canu have lasted so long is because they're so versatile and have so many things implemented. But because they're so versatile and have so many things implemented, there's lots of things that were implemented 5 years ago are still there, but we haven't gone back and tried to see how we could improve it or do better for the current data types.
Right.
So it's kind of a never-ending process of refinement. So in the past, past few weeks, we've been looking at improving some of what we're doing in the best overlap graph with Sergey, and then we're looking at, in the future, I want to replace that with something else. And so Sergey has been looking at some of that as well.
Yeah, and from the perspective of development of different software, I think that we can, first of all, the community is doing a lot of work. Maybe it can do even better job, and we also, I've been quite guilty of that myself. I wasn't, there are probably many parts of the code base that could be released as a separate, libraries, for example, that could facilitate development of other software and prototyping of after the software. But we were not doing it much. And I also don't think that many projects, stemmed up out from the Canu code base as well. But Serge can correct me if it's not true. But nowadays, there are different modules out there, different libraries out there, which actually, so now you don't need to be, a member of SPAdes team to prototype different assembly algorithms, because you can just, get a GFA graph in GFA format from SPAdes with all the additional information that you might need, and then load it into, your own software and do something, then output it, then recompute consensus using some other library. So and I think that's cool. So the more well-maintained, formats and just, modular approach we have, the easier it is for community to experiment. The downside of this is that it might not work as well as if you, for example, use all the available information throughout all the modules. So sometimes the monolithic approach can produce better results, but it weighs on you, and it makes the prototyping harder. Yeah, I don't know. It's messy. I don't know what the best approach is for the software development in bioinformatics.
I mean, that's an important point. I think one of the first kind of projects to try to do this was the Amos package, which was started at the time when Celera assembler was still proprietary. And so this was the modular open source. That's the MOS in Amos, right? It's a modular open source assembly package, which is not much used now because it was targeted primarily to Sanger data and just didn't scale to the Illumina data sets. But the idea there was that, we would implement some useful modules. I say we, I worked a little bit on it when I was in grad school, but it was started before me. And then, you'd have something that can compute overlaps between reads and something that could build a graph from them. And then if you had a clever idea to filter the overlaps or to build the graph differently, you could just try that, and then everything else would be available, right? And that was quite useful. From my personal experience, I can say it was useful because I worked on a scaffolder during my PhD for metagenomic data, and so it was much easier to have that infrastructure that I didn't have to develop all these things from scratch. And more recently, there's been some focus on interchangeable format. Sergey mentioned GFA. There's this PAF format, which is sort of a derivative of the format that the Blazor, the PacBio mapper, put out that Hank standardized with mini-map and mini-ASM. And so, within Canu, we've tried to be conscious of that and put in places where, it has internal databases that are all binary on disk or in memory, but then you can convert them, try to make it so that you can convert them to these formats, which doesn't always translate, like Sergey said, 100%, because it's hard to capture some of that information in some of the formats, but make it easier. So if you wanted to plug in something and replace Canu's overlaps, you could do that. And I know of at least one or two projects that have done that. And Or if you wanted to replace the best overlap graph with something else, you could take the overlaps we computed with all the whatever adjustments we've made to them, corrections to the reads, etc., and use them. But the Vertebrate Genome project has also done this. I'll plug that one format from the Vertebrate Genome project that's on GitHub, as part of the Vertebrate Genome project GitHub organization that tries to define some interchangeable assembly formats exactly because of this reason, because we don't want to make everyone who has an idea for a better piece of the assembler to have to write everything else up and downstream of that, because that's obviously very, very expensive, time-wise and effort-wise, right? So I think having these kind of, I think having the formats be interchangeable is probably the right approach, more so than having kind of a lot of the modules and things implemented in one place like Amos was trying to do, because that gives more freedom, because then, you don't have to play in that one kind of project or playground. As long as you have an intermediate step where you can dump a format that other people can, recognize and read and write out, it makes it easy to have 5 or 10 projects which all have different versions that you can plug and play, and make a third one that combines parts A and B from project 1, C and D from project 2, and F from project 3 in a modular way. And so I think as a community, an assembly community, that's something we want to move towards. And we've kind of slowly been moving there, but it would be nice to get it more usable and more standardized, and get people, facilitate this kind of rapid development, because given how fast sequencing technology is changing, that benefits everyone. The developers, the users, everyone, that we can get faster prototyping, faster development, faster ideas out there.
And so just to close the discussion of SPAdes, Sergey, you promised us a cool story about the name.
About the name.
I always assumed that SP in SPAdes stand for St. Petersburg.
Right. So we try to have some name with SP and A there. So at least SP, but we wanted SPA because assembler. So
right.
So it stands for like St. Petersburg assembler. But also, immediately I had this picture in mind that we would all have the cards, our business cards would have, SPAdes logo on the other side and would look like a card deck. And we actually had those. So a friend of mine, she was doing design back then, and she drew this logo and made a prototype of those cards, and it's been quite cool.
Okay, so now let's return to Canu, and we talked about some of the concepts behind Canu, but just to maybe systematize that information, can you describe the whole Canu pipeline?
Sure. So it's based on this idea of hierarchical assembly. So we try to improve the reads as much as we can before we actually do anything with them. So a standard run of Canu, if you gave it, raw PacBio or nanopore data, the first step is it'll compute all the overlaps between the reads. It will use that to generate new consensus sequences for the read. So it'll actually change each individual read, trim it potentially, split it into multiple ones. But now each read which will have much higher identity quality. And so then in the next step, what we do is use the overlaps, recompute the overlaps again on these new consensus reads, and trim them, and identify chimera or other kind of weird sequencing artifacts that come from these instruments sometimes. And the reason you do this again is because you've changed the read so much in that first correction step that, when you were tolerating like 30% difference in your overlap, you may have missed that there's a couple of bases wrong here. But now that you've improved the read so much, you can say, well, this read has everything is good except the last 4 bases, and you can be much more precise than you would have been. It would have just been washed out at your initial error rate that you have to deal with for this data. And so once you do that, then you've corrected the reads, then you've trimmed them to their best sequences that you believe that are trustworthy. You then compute the overlaps for the last time, build this overlap graph, the best overlap graph to generate layouts. These layouts are initially greedy, as Sergey mentioned, so you just take the longest paths through, eliminate all the reads that you used, take the next longest path through, eliminate all the reads, and do that until you have nothing left. Then you go back and try to fix any mistakes if you see evidence that you took a path where there was a branch, because you're taking a greedy path. You don't have to stop at a branch if you're just going through a greedy walk, right? If you see that you missed a branch that you care about that you likely got wrong, or don't know that you got right, at least don't have any evidence you got it right, you split, and this gives you your contigs. And then the final step in Canu is to then take these layouts of these contigs, actually take all the read sequence, because all these best overlap graphs and all these operations work without ever looking at the read sequences themselves. In the last step, you look at the read sequences, you align them based on the layouts that you've built from your graph. So, where it says read A overlaps read B by this much, so you try to actually align that. And then you align all the reads that were contained that you didn't use in your graph, because we throw out all the contained reads from our graph as a very first step, because the idea is that they don't add anything to your traversal, which is not quite true, as Sergey maybe you can comment on, because he's been thinking about this a lot. But for the most part, it's true, as long as your reads are not too different in length. And then you want to bring them back in at the very end, because they give you information. While they may not give you information about how to walk the graph of the genome, they give you information about what the sequence is at that locus of the genome, right? And so you bring all those in, you align them again, and you call a consensus, which in our case is just a majority variant call, where you build this a DAG from the layout, and then at every position you take the majority path that you have for a vote, and that gives you your final consensus of your contigs. So that's kind of the brief overview of the whole Canu pipeline.
And correct me if I'm wrong, but I think one of the innovations behind Canu was the way you can quickly find overlaps between the reads, and that uses something called min hashing that you worked on. So can you talk about that?
Yeah, that's right. So we had been doing this kind of hierarchical strategy for a while. As I mentioned, when we first modified Celera assembler to do this, we were doing a similar strategy, except we're using Illumina 454 reads. As the reads got better and longer, we actually could do a better job correcting reads with the other noisy reads like PacBio and Nanopore themselves. But we had the problem that we were using old code that was developed for the original Celera assembler and for Sanger length reads to find these overlaps. And so the first time we tried to do a large genome like a Drosophila, a fruit fly genome, it took several 100,000 CPU hours to compute those overlaps, which is obviously... at the time, we were gearing towards trying to do a goat genome, which is a 3 gigabase genome about like a human. And so the Drosophila genome is 150 megabases by comparison. So essentially, the goat genome would have been untractable. And our collaborator, Tim Smith at the USDA, who's been a longtime collaborator on these PacBio projects, called us up and said, like, you guys, you told us we are going to do this goat. How are we going to do this if it's going to take you, millions and millions of CPU hours? Where are you going to get these CPU hours? We said, don't worry, we have an idea. It's going to work. Like, trust us, it'll work out. And so at the time, my friend from undergrad and grad school, Konstantin Berlin, was actually working on protein folding and NMR, nuclear magnetic resonance imaging, and modeling what the protein looks like based on that data. But he had also had some background in numerical analysis, and I had started talking to him about these kind of issues that we were having, and he told me about this min hashing approach, which was proposed by Andre Berdeur for finding similar web pages in searches, so eliminating similar matches. If you, for example, search and you want to tell if these two web pages are largely similar, you obviously can't compare the entire web page to another entire web page. It'll take too long. And so they had this idea of shingles, where you took a subset of words in this document. And the idea is that if the words, what you're going to estimate is this Jaccard similarity, which is how many words are shared out of all the words in these two documents. And the idea for min-hashing is really simple. It's that if two documents are really similar, they should share more words. So if you just randomly selected words from both documents, then you should be more likely to pick the same word from the two documents when they have more similar words than two documents that are not similar. And so the nice thing is that this is independent of the size of the document. And so you could take a fixed size sketch, so a fixed number of words, say 1,000 words, and you would have a good estimate of the similarity of the documents. And so we applied this same idea to overlapping, where we took k-mers now instead of words, and we hashed them all. And then we recorded, for every read, we had these words that we had hashed and selected to represent that word. And then we use that to identify which reads are likely to be similar based on these hashed k-mers. And so that could eliminate a lot of false positive matches, because we could be, we could have various requirements based on, if we model what the similarity of the reads we expect to be, we can estimate how many k-mers we should have hashed in common. And so we can then set thresholds correctly and say, okay, these reads are probably not similar to each other, these reads are, and so we will investigate them further and look in detail at the actual regions where they are similar and how similar they are, and try to estimate that. And so that was the first version of this that was actually predates Celera or Canu, that was part of Celera Assembler. And we published that. And then after that, we started working on other applications of this min-hash idea, out of which the MASH project came out. And this was, comparing and searching database of whole genomes. So if you wanted to say, how similar is Lisi coli to some other random genome you have? And a couple of important things came out of that. One of them is the relation between this Jacquard similarity and the nucleotide identity that people are used to using, right? Because Jacquard is not a very intuitive concept necessarily. And so we had this formula from MASH that let you convert a Jacquard similarity to say this is actually 90% identity, assuming random sequence error, etc., and modeling it as binomial. But we also had this other idea from this document searching, is there's this concept of TF-IDF weighting, term frequency inverse document frequency, which is useful. If you think about the English language, there's a lot of filler words that are not very informative, but they're very, very common, right? So like think articles like the, of, if you know that your text has 10,000 copies of, that doesn't really tell you what it's about. And so this idea, we thought, is very analogous to repeats in genome sequences. And so, if you have this repeat sequence and you have 1,000 copies of it, that doesn't really tell you if two regions of the genome are similar or not, because you expect to find that everywhere anyway. And so we added these two features to MHAP, which is our original min-hashing tool that was part of Celera Assembler. So we added two features. One is that it could use this estimate of identity, which made the filtering and thresholding much easier for us to set and estimate, rather than trying to model the Jacquards like we were doing before. And we added this feature where if we had k-mers that were very common, we could say, we could weigh our, when we were picking this sketch, what is the representative set of k-mers for this read, we would bias ourselves away from picking k-mers that we know are repetitive, and bias ourselves towards k-mers that are unique to those reads, using this TF-IDF idea. And so the nice thing about that is that now you don't have to have a hard filter like a lot of overlappers usually say. If a k-mer occurs more than X times, I'm just never going to look at it. We could look at all k-mers still, but we would just be very unlikely to pick the ones that are very, very repetitive if we had a better option. And so that made the overlapping much faster, and it made it more accurate when you had different levels of coverage in your data set. So if you had some things at high coverage, like a plasmid, and some things at low coverage, like the nuclear genome, you didn't have to worry about how do I set a threshold. If something occurs 10 times in the plasmid and once in the genome, is that the plasmid repetitive or not? you would still find all the overlaps within the plasmid. And so those two main ideas were the changes we made to Canu, which made the mHap step much, much faster as a result. And then, also the cleaning up of the code from Celera assembler. Those were the main things that went into the first Canu publication.
But it was also way before Minimap, right? I think.
Yes. Yeah. So the min hashing, the mHap idea was one of the first applications of this min hash kind of sketching idea to bioinformatics. There was some limited work, I think in the late 90s, early 2000s, I guess, where people had used it for some similarity searching, but it had been kind of forgotten and not used in bioinformatics for a long time.
Right.
Before.
No, I meant mostly that at that point there was actually no other good way to compute the overlap between long noisy reads, right? So nowadays everybody can take a Minimap and it will produce reasonable results, although there are some catches with respect to assembling complex genomes, but for general use it's fine. But at that moment there was nothing, right?
Yeah, there was nothing. It predates both Minimap and DLiner, which is the tool that Gene Myers developed, which also was trying to solve the same problem of how do you find all these overlaps between reads fast enough to be able to use them for assembly. And so it was kind of our paper where we used this tool was the first one to show a whole genome assembly of a mammal genome like a human. Was now possible from the long read data, which before would have taken, millions of CPU hours. It would have been impractical really to do it. Yeah, and one of the other key ideas that came out of that, which is, you reminded me because you brought up Minimap, is that you don't actually, before that, all the overlapping had been focused on finding an actual alignment, whereas, you do a dynamic programming or something to actually find a base correspondence between reads that you're looking at. And so we said in the original mHap paper that we can estimate that this will have a good alignment anyway. We don't really need to do the full alignment. We can tell you this is pretty good. And this one is not very good, right? And we can discriminate the levels of good and bad without having that step. And so we essentially threw out this expensive step without sacrificing the quality of the overlaps that you're finding. And this is similar to what Minimap is doing in Mini-ASM, that they throw out that information as well, all the way through the assembly, actually, right? And so that idea has gained some traction, and it does actually seem to work, that you can estimate from these kind of K-mer statistics, you can estimate the information that you would be getting from a dynamic programming and be pretty accurate about saying what is a good overlap, what is not a good overlap.
Right, and another, I don't think you mentioned MeshMap, right? Which is the latest installment of this idea from our lab, led by Chirag Jain.
Yep.
Right, and Roman, if your listeners are not familiar with this work, it's pretty cool, and it's also a map, it supports local alignments, I think, of so if you have your genome and you're interested in aligning all of its 5K windows to the entire RefSeq database, for example, it can do that in reasonable amount of time and memory. And also they showed how you can do pairwise comparison, pairwise ANI, which is average nucleotide identity index computation across 90,000 different bacterial genomes, and then show whether there actually is a species boundary at 95% ANI. But yeah, so it makes this it makes alignment of big queries to large databases fast, and it also doesn't do it based on nucleotide by nucleotide comparison, but it uses this sketches and gives you correct result with essentially high probability, right? And you can estimate the probability that you missed something. So yeah, it's pretty cool work. So check it out.
Yeah, I mean the min hashing has been a quite cool thing that we've applied, starting with the assembly and then mash and then mash map. I mean it's a really simple, intuitive idea, but it's extremely versatile in the kind of problems it can be applied to, especially with some of these modifications that actually Chirag Jain, who is the author of MashMap and this FastANI tools that Sergey mentioned, and is currently a postdoc in our lab after being a summer student. When he developed all these tools, he's looking at ways that you can apply some of these TF-IDF ideas that we used in assembly to mapping as well. So how do you avoid, how do you map to repeat regions in the genome correctly? Given that the typical approach is just to throw all of them away and not try to map reads where they have these repeats, can we actually do better than that and try to map in those repeat regions? So yeah, I think it's a very cool and versatile idea.
Yeah, but interestingly, in Canu we go to a completely different direction, right? So no sketches will be enough for us. We are going base by base and looking at the highest resolution possible that we can achieve. So yeah, I would love to use some sketches, but unfortunately, I wasn't able to yet.
This concludes the first part of our conversation with Sergey Nurk and Sergey Koren. In the next episode, you'll hear the rest of this conversation where we talk specifically about hi-fi reads and high Canu, so stay tuned.